Hello everyone, today we're going to talk about the basics of protein ligand docking. So what is docking? So the docking means something is being docked. So when you think about a ship, and ship docks to a harbor. So the docking means that two objects are connected to one, and so the docking is used as a term, in protein science, and usually the docking means that a ligand, a small molecule, an organic molecule is bound to a protein, or two separate proteins are bound together. So the docking means that the docking simulation or the docking modeling means that they to find the best matching, to find the best structure between two molecules. So let's get into more details. So the definition of protein ligand docking is that the protein ligand docking is one of the molecular modeling techniques. And the goal of protein ligand docking is to predict the position and the orientation of a ligand, a small molecule, so when it is bound to a protein receptor or an enzyme. So in other words, the protein ligand docking means that the finding the right conformation, finding the global Gibbs free energy minimum conformation of a ligand when it is bound to its receptor. Because all chemical reactions spontaneously happens to be the global minimum of Gibbs free energy, right? So then, why is protein ligand docking important? Because many proteins are regulated, in other words, controlled by the binding and unbinding event of ligands, so the small molecules. Therefore, they play important roles in drug discovery. So actually, most drugs we take every day are actually ligands, and when those drugs are observed to your body, then they are observed to the cells, and they are observed to the cells, and somehow they are bound to their target proteins. And once they are bound with their target proteins, and the activities of proteins are controlled or regulated by that binding event. So the activity of protein can be enhanced, or the activity of a protein can be, prevented or suppressed. So by using protein ligand docking, the drug candidates can be efficiently searched from chemical databases. So as we talked in the last class, the drug discovery process is a very expensive and time-consuming process, and especially because you have to find a right molecule that binds with your target protein among thousands or tens of thousands of candidate molecules. If we can find the right protein ligand complex structure, then we can design new drugs much more efficiently, right? So the goal of protein ligand docking is that to find the correct structure of protein ligand complexes, then so that we can find new drugs or drug candidates more efficiently. But protein ligand docking is not trivial. It's not straightforward. Then why is this so? The goal of protein ligand docking is to find the, as I mentioned, to find the right conformation, so the right bound structure. But in order to find the right conformation, actually, so we have to test multiple possible conformations. In other words, it is called as sampling. So it means that we have to sample large conformational space of ligands. So in other words, so we have to test multiple, so many conformations, right? So let's take a look at the example on the right side. So if you take a look at the examples on the right side, so they are showing some docking results of small molecule and a protein and a receptor. So there are, many small molecules and not many. So here the yellow, green, and red means so the different correspond to different conformations of a ligand. So one ligand can have multiple conformations, so which means that they can bind with a protein in a different ways. Why is it so? Because a ligand is in general flexible. So as you know, so ligand is flexible. As you have studied, as you have learned from organic chemistry class, so single bonds are rotatable, right? So the single bonds can be rotated. So a ligand can have multiple conformations when they have, many single bonds. So based on, multiple conformations, so the ligand, so based on its multiple possible conformations, a ligand can adapt multiple different binding poses. The bound conformations. So on the bottom picture, so as you can see that a ligand is bound with a protein in many different conformations, and so for efficient and rational drug design, so we need to know how a protein and a ligand interact accurately, right? So that is the goal of protein-ligand docking. So this is the overview of the protein-ligand docking problem. Okay. And also the second reason why protein-ligand docking is challenging is that so it is hard to know, it is hard to measure, or it is hard to score the strengths of protein-ligand interactions. So here I mentioned scoring, but you can say it as measuring or estimating, something like that. So in other words, it is predicting the binding affinity between protein and ligand, right? So which means that if we write down it as a chemical reaction, that the protein-ligand binding reaction can be written like that, and the binding affinity corresponds to the free energy change of this following reaction. So the free energy change corresponds to the binding affinity. So the free energy change of this reaction corresponds to the binding affinity. So estimating, measuring protein-ligand interaction accurately is very important for finding true binders. So which means that so let's think about so if we take a look at the example on the right side, and so many ligands, there are many possible ligands that can bind to this protein. but we don't know, so which one will actually bound. We don't know, which one will actually bind. So whether this will bind or not. Actually, so before we do, experiment, we don't know, whether this ligand will bind or not. But so if we actually do some, biochemical experiment, then we can, discriminate. We can identify, so which ligand will actually bind to a protein. But doing that, biochemical experiment is quite expensive and time-consuming and labor-intensive also. So if we can predict, accurately whether this ligand will bind with this protein, without doing experiments, then it'll, save us a lot of time and effort to find, or to screen, real, drug candidates, promising drug candidates. So that is the goal of protein ligand docking. But in order to do that, so we need some kind of function to measure, the strengths, the strengths of interaction between protein and each ligand. Okay. So the question is, so which one will bind? So which one will not bind, and which ligand will bind most strongly? So among, all possible, binders, all possible ligands, so we also need to identify the strongestly binding, ligands to develop efficient drugs. If ligand is not interacting with the protein, very effectively or strongly, then, it will not be an effective drug. Otherwise, so we have to take it, we have to take that drug, in a large amount, to be effective, right? So let's take a look at some example of protein ligand docking. So the actual example here we're going to take a look is a HIV protease. So HIV is, as you know, it's a kind of the virus. So it's a AIDS virus, right? And protease means that, as I mentioned, so AIDS means cut. So AIDS means an enzyme that cuts something. So they are called A's, and protease means that an enzyme that cuts proteins. Okay. So if you take a look at the life cycle of an HIV virus, then HIV virus is fused with the host cell, and the RNA of HIV is inserted into a host cell, and RNA is released. And that that is bound with reverse transcriptase, and so it is transcripted to a DNA, and its virus DNA is actually inserted to the host DNA, and host cell just transcribes this DNA into a protein. Okay, translates this DNA to protein, and by doing that so new viral RNAs and new viral proteins are synthesized, and they form a virus, and they leave from the host cell. Okay, the virus is released at the end. And so usually so the RNA HIV virus transcripts its DNA using the host cell's ribosomes. So usually so once it is formed, once protein is formed, long protein chain is formed, and they are cut. Here, so they are cut into, several pieces. A long protein chain must be cut to form, separate, functioning proteins. So the HIV protease plays a role here. So the HIV protease comes. So this is the, whole long chain, but HIV protease comes and it identifies some cleavage sites and it cuts some necessary points. So the HIV protease cuts a long protein chain. Once this long protein chain is cut properly, then each protein works as expected. Okay. So the HIV protease cuts a long protein chain into multiple functioning chains. Okay. So otherwise, so if that long protein chain is not cut properly, then long protein chain itself does not work. It does not function properly. So once they are cut properly, so each protein works as expected. So they plays their roles. So this is the structure of HIV protease. Its PDB ID is 1D4S, and here so you see so there is a hole. There is a hole in the middle of the protein. So this is actually the active site of this HIV protease. Okay. So the long protein chain, peptide chain, goes into this hole, and protease cuts those pre-assigned cleavage sites. Okay, so based on that idea, so if we block, this active site, then the peptide chain may not be inserted into this hole, into active site, and it may not be able to be cut properly. So which means that the HIV virus cannot be reproduced, and the HIV virus cannot, work. So that was the one of the idea. And so here, so on the picture right side, so this is the structure of the HIV protease docked with, combined with or bound with a ligand, bound with its inhibitor. So inhibitor means that so this ligand inhibits, prevents this protein from working. Okay, so its PDB ID is 1EBY. So if you take a look at the 3D structure of this crystal structure, then since this active site is blocked, and so the protein chain cannot go into this active site, and this ligand is actually prevents the reproduction of HIV protease, and eventually prevents HIV from reproduced. So basically so this becomes the suppressor of HIV virus, or in other words, suppressor or the cure of AIDS virus. So in these days, actually, the HIV AIDS is a manageable disease. So even though a person have an HIV virus, so it cannot be completely cured, but still, so it can be managed to a certain level. So if you take proper medicine for life, then so you can live for many years. So here the important concept is that this a site where reaction occurs or a ligands bind is called an active site or a binding site. Okay, so the active site of an enzyme means that the actual, site, actual, substructure where chemical or biological reactions happen. Okay?